All right. If you want to start your Labor Day weekend, we have to get it started. And actually, I gave a seminar today at noontime, so this will be my 4th hour to speak this afternoon. So I apologize if my voice is getting away a little bit from me. Okay. All right. So we talk about the BAM file, and just barely through that a little bit, but you can see my point is. And my point is not telling you which bit means what. It's really there's a lot of information are being impacted in this, packed into this file format. There is a tutorial you can read, and if you really need to do anything, and there's a code that you probably need to write a little bit to get the information out. Another couple of concepts I thought is important, so I want to mention, is the first one is for this one. So let me see. Yeah. So for this one, it's very simple. It's a 7, 9, or whatever. This is basically the genomic locations of where the read starts in the reference genome. Nothing fancy, but they made it really fancy now because this can be very difficult to interpret in the way that this is there are 2 different systems of this. So one is called a one-base coordinate system. The other one is called zero-base coordinate system. They are different, okay? So when you do the one-base coordinate system, is the first base of the sequence is 1. This is pretty straightforward in a sense, right? And then a region, if I say I'm looking into the position, the nucleotide number 3 to 7, so basically, you say from 3 to 7, and that is your nucleotide. Okay? So that is a one-base coordinate system. And actually, some GFF Wiggle files in the UCSC genome browser, they all use one-base coordinate system. But just to make sure to confuse you enough, there is a zero-base system as well. So what that means is the first base sequence is 0. And then the second base is 1. Okay? Actually, what this gives you is not, you should point your arrow not on the nucleotide, but rather on the gap in between. So when you say it's 1, that means the first gap. And then the second, the third. That's why that if you are trying to refer to nucleotide number 3 to 7, you will have to make this is like this from the second gap here, which is number 3 to number 7. Okay? So nothing fancy. It's just annoying. And it's just a standard you have to be aware of. And we have been in the past that generate some of the data, and we need to write a code to say, let's load the data into the UCSC genome browser or IGV, which I will talk about later today. And then when we do that, we see every single nucleotide shift 1 base. And that is because we didn't pay too much attention to this. When you do the variant call, when you do many stuff, and this part can be your major source of error if you do not pay attention. And also, the strange part is the BAM file is actually zero-base started. So whatever it is, just pay attention and making sure that you fully understand this. Okay? So then, yes?
Right. So what's 37 here?
37 is the position, the nucleotide position in the reference.
And that's based on the one-base coordinate system.
This one, if this is a SAM file, that will be based on 1 base.
Okay. So it should specify based on the file.
Right, exactly. So the next one is going to be cigar strain. And this is another thing that become very interesting. So you're seeing here is 8M, 2I, 4M, 1D, 3M. Okay, so So this is a cigar strain. Actually, this tells you how the alignment is. Why? Let's take a look at it. Did I have? Yeah, I blew up this a little bit. Okay. By the way, here is M means match or mismatch. I means insertion, deletion, hard clipping, and soft clipping. So Chi and Jun, do you know what the difference between the hard clipping and soft clipping? I only know that there is something hanging around here that is the clipping. I never figure out, but that's what we're going to talk about later. And then P is the padding and the N is the escaping. So let's get back into it to see what really this means. I'm making this bigger. Very, very important. So the first one is, so this is the reference genome. This is genomic location coordinates. This is your sequencing race, TET or whatever it is, okay? And this is the cigar strain of this one is like this. So what that means is 8M tells you first start from 8 base that is perfectly matching with the reference genome. Then there is a 2 nucleotide that you don't have that in the reference genome. So that's why there's a 2I in here, 2 base insertion. And then you have another full base matching. So you have 4M. And then another 1 base deletion, because you have this position as a G, as a reference, and now you don't have it. So 1 base deletion. And then ended with 3 base of mapping. Okay, so this cigar strain actually tells you how the mapping is, how many gaps are in the middle, how many insertion deletions, and tell you those type of information. That makes sense? Okay, so basically that is what the cigar strain is. And again, there is a lot of information regarding what our different letters means. And that it's not important, just go to figure out. If you need to know, there's a dictionary you need to check. But this will tell you exactly how the alignment goes. Okay, so the column 9 here in this BAM file is going to be inferred fragment size. Okay, so now I'm coming back to this particular example. First of all, this is only for paired end rates. So if it will be written as 0 if it's a single end, okay? And you see here, a lot of them are zeros. Those are single end rates. But for these two, 39 and minus 39, those are coming from the same read. So these are the paired end. Does that make sense? Okay, so this is only for the paired end rates. The general idea of it is from these 2 ends, figure out the length of the initial DNA fragments you're sequencing. Very understandable. What you need to do is, if you made a calculation, this start from 7 and this end at 45, and then you see if this is a 0 base, and then you know there's a 39 nucleotide in between. That's why you are putting 39 here. And the leftmost race, you put a positive number, and the rightmost race, you put a negative number. That is what it is. That makes sense? Okay? So that is also included in the BAM files. Where are we? And on this. So, and there are many different optional fields in the BAM file, which you will basically give the format like NMI2, and that is actually NM will have a definition, and the I is the data type, which is an integer, and value is 2. So NM is the number of mismatches and things like this. It's called edit distance. But this option afterwards is really optional. You can put into the BAM file or not. And if you need to know that, or if you want to write your own BAM file to put into it, and then you can check this definition to make sense of it. Okay? So that is basically how this part is being defined. This is the band file. There's other optional information. Again, it really doesn't matter exactly what that means. It means what I need to know to is there are information in that. There is a manual you can check, and you should be aware of that. Okay, and then BAM is the binary format of the SAM files, and there is another file called a BAI that is the indexing file of the BAM, and sometimes they come with together, and then we in many cases can help us to speed up the calculation. And BAM must be sorted by the reference ID and then the leftmost coordinate before indexing. So, but anyway, those are some details. Again, the information of the BAM file, this is one of the major contributions of the Southern Genomes Project, actually, and it's one of the standards for this, okay? So now I'm going to quickly go through the last one, which is the VCF files. And this VCF files is basically the variant information. There are a few variabilities of this. Similar as a BAM file, it's also first developed in the Southern Genomes Project, and then it's a standard format. It has a lot of information in that. And usually stored in a compressed manner and can be indexed for faster access. So for the VCF file, there are two sections, the same as the BAM file. One is the header, the other one is the body part of it. There are a few things I want you to pay attention to. I mean, most of the part is pretty self-explanatory, okay? But I want you to see is this is the information in the BAM file. And then you can see there's a chromosome number, position, ID, which is the RS number. Those are the database of the variance entries being registered there. And there's a reference genome and alternative genome. So those are very, very self-explanatory, very, very straightforward, okay? And, but I want to focus on this a little bit. And there's one point I want to make. So for SNP, very simple. So you see, if this is your reference is ACGT, your own sequence is ATGT, the second nucleotide is different. So the position will be 2, and then the reference is going to be C, and the alternative is T. So this is what that means. Very straightforward, nothing fancy about it. But for the insertion, that will start become interesting. So you see this ACGT, and in your sequence is ACTGT, meaning there's a T being inserted. Instead of saying this is the third position, you actually say position number 2, and the reference is C, alternative is CT. This is the way that you tell the system this is the insertion, and this is a kind of added one number. So you don't really tell it's number 3. The same thing as a deletion. If the ACGT and in your sequence is A, but CG is gone, it's only T. And then your position here is going to be 1, okay, instead of 2. And your reference is ACG, your alternative is A. So this is how that works. And this is a complex events. There's one more T here, but it's a pretty self-explanatory on this. Does that make sense? So just one small detail I want you to pay attention to. And the next part is going to be variant quality. So we'll talk about how to calculate a variant quality in a few lectures later. But you can see this is also the same Fred score calculation of the probability you are making mistake, period. So it's that simple. Meaning we really want the quality to be as high as possible, okay? And there is a filtering here, which is says most of them says pass, but there's one says Q10. So if it's not a pass, that means it's failed. and filled by what? By Q10. So what that really means is this Q10 is actually being defined in the header files. So what that tells you is in the header file, so sometimes you see even more than one Q10 and S50 there. So what in the header file, there is two rows. One is a filter Q10 quality below 10, and S50 is less than 50% of the samples have data. So if you define in this way in the header, and then when you say it's Q10, that tells you this position failed the quality, didn't pass the quality checking because of this particular filter is not being met. That makes sense? So you need to look into in conjunction of this together with the header files. And the next part is the information part, which really gives you the information for this particular SNP, okay? And so you can see there's a NS is equal to 3, DP is 14, AF is 0.5, and so on and so forth. So what this really means is there are many predefined fields in this VCF file. For example, NS is the number of samples with data is equal to 3. DP is a combined depth across samples is 14, and so on and so forth. So these basically the text is defined in the VCF definition, and then it's just gave you the information for this particular location. Does that make sense? So you can check it. It's like a dictionary, and there's a manual of VCF files. You find out which individual tag and what the number will associate that means. Okay, so the next couple of one is also very important. And so for this particular information column, this is for this particular location. This is genomic variant location. It has nothing to do with individual samples, because you can possibly put 100 people's VCF file into this, the variant information into this one VCF file. So it's not like a one VCF file for one patient. No, you can download one VC file for all the 1,000 genomes data, 1,000 or 2,500 individuals. They're all in there. So this part tells you the information for this variant location. And then after that, each individual column gives you for that particular patient what the information would look like. So if you see this part is you got a predefined field, what these individual numbers mean, and they have been having different format, like GT. The first one is GT, that stands for genotype. So you can see here is the first one is 0-0. That means it's a homozygous reference. And this one is 1-0, it's a heterozygous alternative. This is 1-1, that's a homozygous variance. But every single first position of those numbers will tell you about the genotype. And then the second one is DP, and then which you can look at. DP is the read depth, how many reads cover this particular location. And for this particular one is 1, and for this one is 8, for this one is 5, and so on and so forth. So basically, it's more like this is a format, it's more like a dictionary, and then afterwards will be the data file. So VCF file, what I'm telling you is very simple, like a SNPs or small indels, but a VCF file can be very complicated, hosting many other type of structure variations as well. And we don't really need to fully understand this at this point. But if your research leads you into this area, you can try to look into it. So basically, this structure variation, the VCF file will look like this. And actually, they can be more complicated as well in their original VCF file papers. Okay, so I think I covered most of the information I need to cover for this lecture. And there are many more slides in the notes, which is I want you to go through by yourself in terms of the genome browser, what that is, and play with it, and things like this, okay? And some of them I basically steal from genome browser website itself and to generate it. So I'm not going to go through this. I think it's very self-explanatory. However, what I'm going to do is there is one thing I need to point out. Let me see where that is. Okay. So where is my slides? All right, I can still continue play it. Hold on a second. So in one of the slides I have is actually I provided some of the examples of the real data. Okay, so let me go to the genome browser itself. This become really annoying. Okay, so let's go to genome browser.ucic.edu. And this is the initial page. And then if we want to go to human genome, there are many different genome builds. Again, I'm not going to go detail of this, but you can basically go there. So let me clean up this a little bit. So most likely, this is going to be what you see that after you look at the genome browser. And again, there's each row is one type of annotation, and this will tell you the gene information where they are, but you can review the slides. But the point I want to make is this is a tool that is not only helping you to visualize the annotation, but also you can load your own data to visualize your own BAM files. Okay, so what I'm going to do is very quickly demonstrate how to do that. Let me change the setting a little bit. Make it more visible. All right, it's a little too small, I think. Make it a little bigger, I guess. All right, so one thing you can see that this is one particular gene and where the exons are, introns are, and things like that. But you can go back to see there are many different databases, and then if you turn something on, and it will tell you where the gene is, and there are many different annotations. But if you look at this, there is several different buttons at the bottom. One of the buttons is Add Custom Tracks. Okay, so if I click this one, and then this is the place that you can add your own custom tracks. Again, you can read all those documentations, and those are all different type of files you can load in. And then if we go to our slides here. And in this page, and I made this, actually one of my assistants made this. You can paste and copy this, and you can make this file by yourself, okay? And then you paste and copy that part into this section, and then you do submit. So now you are having 2 more tracks being submitted into it. And then you click go. So now you see that there are 2 custom tracks here. And they are both our samples just being submitted. And then if I change this setting into, for example, squish, and then refresh. So now you're seeing these are our sequencing data being aligned here. So what I provided to the software is the BAM file. It's actually a link to the BAM file, okay? So let me go through this and I'll tell you how this works. So you can see that this is an original annotation. So there's an exon here. And then you see that there are many reads here. And then in each one of this, if you click, and then it actually goes to this read. And it tells you what is the read name, chromosome location, beginning and end, and many other information, including the sequencing quantities towards the end. And this is where the alignment occurred, okay? And then we go back, for example. And then if you look at a read like this, it's the same thing, okay? So now let's take a look at where's the cigar string of this, somewhere. Here we go. The cigar string. Here is 38M. It's not very clear. It's a 38M, 796N, and 36M and 2S. So 38M, that means the first 38 nucleotide of this read map to the genome. And then there is a 796 nucleotide gap in the middle. And then there is another 36 nucleotide mapped. So let's go back to this. And so this is one of the reads I collected. This is very clear. This is RNA sequencing. And then because we sequence the RNA molecule, it's a mature RNA sequencing. So one end is in one exon, the other end in the other exon, okay? So you can play with this. And actually this tool is quite good. So if I zoom in here, you can do that. And you see that there is all those detailed information, and there is one part which is here. Somehow there is a variance there. I cannot see. Maybe if I turn this into, don't do that. So if I click this in, where is it? It's here. In the reference genome, it's C, and the alternative is T. So if you look at this particular data sets, and every single nucleotide reads that this is the same variance being identified. This really tells you that this particular location, this person has a nucleotide, a SNP in this location. So the point here is, which we'll go through this in the future as well. The point of this part is you use the genome browser. If you do it properly, you can really visualize your data pretty well. So let me turn back to our slides. Okay, so So what I supplied to the genome browser is some of the information about this track and which reference genome it is, and also provided a link by putting our own data on that, our own BAM file on that. So this is a HTTP link that is service hosted here at IU. Here's how it works, okay? So we are visiting a site in the UCSC genome browser. It's Universal California, Santa Cruz, okay? And then we say, for this particular gene data, I want to see this region. I send my command through the browser to UCSC genome browser in California. And then they will look at which location that I'm interested in, come back to the IU server in our HTTP server, and then to specifically query that part of information, send it back to California, and display that in the information, and then send it back to our browser and to visualize these tracks. So in a sense that we do not need to upload our data to UCSC. These are going to be gigabits of size of data. We don't need to do that. We just provide a link to the UCSC genome browsers. It's very, very fast process, and the data is with us, and we can still visualize using their infrastructure. Okay? So I think that's all I want to talk about, and Sean, you're up. I'm going to take a break. Any questions so far?
How do you interpret the data? Like I saw the example that you showed that there was one variant, but
right.
There are like so many sequences there.
Right. And hold on, let me put this up and then answer a question. I cannot do two things at the same time. Sean, is this the one?
It should be Indiana University Supercomputing. Yep.
Okay. Yeah. So some come back to this. So what was the question again?
The long stretch of sequences that you're looking at.
Yeah.
How do you analyze the data?
Well, so if you really want to do this type of analysis, you want to identify all the variants in this RNA sequencing data, which we will talk about in the later. We will need to generate a VCF file, and by finding out all the genomic locations that has a variant. And then you come back to individual loci. You cannot possibly go through everything. Okay? Sorry, I'm over time a little bit.
No, you're fine. I'll just a lot of it they can just read about.
Yeah, sure. You can move your slides down.
I don't get mic'd up very often. Can you all hear me? Junwan, can you hear me back then? Okay. I just wanted to take a few minutes and introduce you to some of the environments that you're going to be exposed to and that IU has provided for you. You're in a computationally rich environment here at IU. And I really want you to see some of the tools that you're going to use as we move through this semester. And also as you do your dissertations or you do your postdoctoral work, some of the environments that IU will permit you to do large-scale computation in. So as I know most of you have probably just gone to the bottom of the page right here, don't worry about it. There's no homework yet, okay? What I want you to understand is where we're going based on what Dr. Liu just presented to you today. And as we move into more of the DNA sequencing, some of the hands-on tools that I'm going to give you to provide your own computation, this way you can see how these theories are applied and what the output really looks like in your own hands. A few of these things you may already know. A few of them you may need introduced to you. But we'll just kind of move along efficiently. And let's see, where's my... Oh, okay, there was one slide that I had added that I didn't give you on the new one. The most important thing you need to know how to do is access the computation area. So as far as I understand this course, there's some folks from informatics majors, and there's folks that have biology sides. You all need to be able to access the computational resources. And I believe it's access.iu.edu. Okay. I will provide that slide to you guys just so you know. What I want to remind you of too is one, we're at Friday after 5 o'clock. Two, Monday is Labor Day. So some of the UITS folks, university IT people, may not, if you apply for it today, it may happen a little bit slower because of the holiday. But be aware, even though it says homework assignment on the slide, it's not there yet. You have time to get into the computational arena. So I believe it's access.iu.edu. How many of you are familiar with knowledge base? Have you ever used knowledge base? A few of you have. That's wonderful. It is a key as you move forward because each of the environments that you're going to work in that I'm going to introduce today have pages that will give you hints how to submit jobs and operate in some of the languages that you'll need for those different environments. So I highly recommend IU Knowledge Base. It's kb.iu.edu. And once you get in there, there's a search bar. So if you search High Performance Compute Cluster or KARST, you're going to get a page on system overview and job submissions and exactly the type of things that you're going to need to be successful in computation. Okay? As you go through this, what I want you to remember is Google is one of your best friends that you may ever have as you try and write code, run data, analyze data. People that have degrees in bioinformatics or data science or computer science, use Google. Okay, we all get stuck. There are forums, there are boards, you can ask questions. But if you don't start out asking Google the first question, how do I run BWA? Well, it will tell you. There will be hints that other people have posted. Make Google your best friend as we move along this semester. And also, use the knowledge base. As we move into DNA sequencing, which is some of the lectures that we're going to go to, another thing that will help you a lot is provided from the Broad Institute at Harvard. It's called the GATK Best Practices Guide. GATK stands for Genome Accessory Toolkit. It will give you hints and some of the best practices to get the highest quality data that you're trying to get. So you have Google, you have the knowledge base here from IU, and you also have the Best Practices Guide. You will, however you set up your system when you try and access the knowledge base, it'll give you the dual, you may get your sign in and then get a phone call or a text. That's how you'll get into it. It is critical to make this as commonsensical as possible because as you watched Dr. Liu tell you about the alignments, this is intricate. Okay, this is a field. This is an absolute science. So use as many of the tools that you have available to you. The bottom line, the HPCC versus the Cray is a high-performance compute cluster versus a supercomputer. And we have both of those available for you, and I want you to see some of the differences. One of the compute environments here that's very commonly used is called KARST. It is a high-performance compute cluster. It was introduced in 2011, the high-performance compute cluster, and it's set up on a node system. The one here has about 16 different nodes plus a head node. So when you submit a job to it, it lets you do parallel sequencing for these massive files. So it doesn't do just one nucleotide at a time. It doesn't do just one read at a time. It'll divide your job into large nodes into different places. You may get 2 nodes. You may get 4 nodes. You're going to go into a queue, so it may take you a while to get through this. And as Dr. Liu mentioned last week, once you submit a job to KARST or once you submit a job to Big Red, it may not run right away. You may have to wait a little bit. And to give you an idea, when we do whole genome sequencing, those jobs, once we get into the queue, can run for 72 to 96 hours. So it could be 3, 5, 7 days. So be patient. Be patient with your data and always check the status of the run that you've submitted. Can anybody think of why you'd want to keep checking the run? They'll stop. There are points when corruption could happen, a bug could happen, a mechanical failure could happen at KARST. They could be doing an upgrade and it'll stop your flow. So always watch your workflows and use them as you go. Parallelization is the key. This is big data. Okay, these data files can be terabytes. They're huge. So you need to break them up as to many nodes as possible to increase the speed time to get your output back. Because as Dr. Liu said today, the most important thing that comes out of this is biological sense. Okay? You want to have your data coming back in a way that is trustworthy, it's reliable, it's repeatable. Someone else can take your files, align them, and get the same results, okay? That may or may not happen depending on which human genome they're aligning to. Some folks still believe in HG19. Some people solely go with HG38. So you may see some differences. But the key to it is repeatability and reliability. So parallelization makes some of that possible. I'm not going to really talk to you too much about, just because of time and it's Friday and you all want to get home, I'm not going to go into Hadoop and architecture. These are considered supercomputers, even though you're about to really see a supercomputer. IU has an unbelievably great system built by the Cray Corporation, which was developed in 1972. Their whole goal was to try and compete with IBM and Bell Labs. They were the big computational people in the 70s and the 80s. And when they went through a restructuring, they decided that supercomputers were the way to go. These are used at Oak Ridge National Labs. They're used at Bern in Switzerland at the Hadron Collider. They're used by the US Department of Energy. Massive, massive computers. So if you see the bottom, this is from Cray. What we have here is called Big Red 2. It's Titan. They're saying they could scale our computer here up to 500,000 processors if we had the money. Okay, it all takes money. Computation is not cheap. So Big Red 2, this is right off of the IU knowledge base. Big Red 2 is among the world's fastest research computers. Keep that in mind. This is one of the fastest research computers currently available. So IU has put you in a very rich data position to run large data. The two Cray computers, XE6 and XK7, are two separate computers, and they're joined and interfaced by a system that was developed by Cray called Gemini. This is why it's called Big Red 2. It's not one, it's two. Two supercomputers linked together. And look at this, there's over 1,000 compute nodes. We had 16 on Karst with the HPCC, okay? Over 1,000 nodes, 21,000 processing cores. We're talking high speed, big data. So a lot of RAM that they throw at it. And I'm going to hold off on this because what I want to do is give you a real demonstration when I come back in a couple of weeks to show you how to go hands-ons with these systems, okay? There are commands that we can put into the system that will tell you all of the softwares that are available, the versions of it. And so it simplifies some of the command and the coding that you have to do by already knowing what's available on the system. Some things you may have to download, some things are already provided for you. But that's a way to find out how it's done. There's another segregated computer, it's called Big Red 2 Plus. How many of you guys have ever heard of the Grand Challenge? Have you heard of this from the chancellor of the university? So there are some avenues that the university wants to address. And the School of Medicine has been charged with taking on a precision medicine initiative to cure one childhood cancer. That's part of a grand challenge, okay? And so this system right here is put aside to run specifically data tied to grand challenges for precision medicine. It's a high-performance supercomputer, and getting access is very difficult. Your project has to go through an application process. But know, if you're working in some of these labs, say for cancer or other things, you may be dealing with Big Red 2 Plus, okay? So that's three systems we have currently. We have Karst, which is a high-performance compute cluster. We have Big Red 2, which is a supercomputer. And we have Big Red 2 Plus, which is set aside for precision medicine and the grand challenge. Here's a little hint that I'm going to give you if you are working in Karst. You have 16 compute nodes. Okay, they're broken up into two parts. One's a data registry, one's a computational side, and then there's a head node on it. When you submit your job to Karst, do not put your job on the head node. If you do, you won't make friends with the system administrator, because what happens is, based on the size of your job, the head node typically distributes everything to the other nodes. If you assign it to the head node, everything else stops until your job has completely run. So if you're running a job that takes 7 days and everyone else is in a queue and you're on the head node, you didn't make friends, okay? So just be aware that there are some folks that are going to take that a little bit personally. We'll talk more about that once we go into looking at the system and I give you a demonstration of it. Karst is a little bit smaller than what you saw with Big Red 2, right? So there's 228 general access nodes. 28 condominium nodes, and then 16 dedicated nodes. Computation is expensive. Okay, it's not free, it's not cheap. And so the way this works, the reason these things are called condominium nodes is IU Bloomington set this computer up. So if your department needs specific nodes to run a major project, for example, you're doing some things that DNA ancestry or registry that you want to do, and you want to run some files, you can block off certain nodes and they'll charge your department a fee. But as soon as your project is over, you'll take ownership of that node for the project. And then as soon as it's over, the node goes back out into distribution. So it's almost like a timeshare. So it's a condominium node. Now, if it's a dedicated node, this is when the department has paid for a node. They have 1 to 2 nodes. It's their nodes, and they run it. And that's set up in a separate way just to make sure that these can be dedicated and won't be in competition with the condominium nodes. So there are many, many resources here and many ways for you to use this system to get your data run and to get your output back. If you're signing in to use Karst, okay, for example, for me, it would be ccourt@karst.uwitz.iu.edu, and then that'll put you in the environment. Do any of you have Karst Desktop? Do you have access to Karst Desktop? I use that all the time. The joy of it is it gives you Karst, but it also gives you DC2 Scratch right in the same window. So you can move back and forth between environments. You can move files pretty readily. Look into Karst Desktop, talk to your PIs about it, and see if they're interested in getting it for you. So Linux is a working environment, okay? It's the backbone for everything that I've just shown you. So the commands, the language is typically C or C++ that it's written in. And we go back to Google, which is your best friend when you're doing computation. If you type in Linux commands, I've given you 2 links to pages that will give you a sheet that you can print off or just store to your desk that will give you basic Linux commands. How to operate a system, how to copy, how to quit, okay? Simple things that usually trip people up. What you'll find in computation, it's very hard, but usually the mistakes are quite simple. There's an extra space, or I don't know what directory I'm working in. So if you type in, if you're in your Linux environment, you type pwd, it tells you your present working directory, okay? So these are sheets that will give you cheat reasons to just work within the environment. So when you hear about Linux, remember, it is the operating system, the working environment. The language is typically C or C++. So a lot of people say, oh, I'm in Linux. Well, it's an environment, okay? A language is different. So again, this is your present working directory, and I will go hands-on and show you at the next round. I will show you what happens when you type in ls to a Linux system. It lists everything that's in your directory, okay? And then there's some other codes. Now, if we're in DC2, these 2 right here are going to be a problem, and I'll tell you about those next. I won't cover these because Dr. Liu did me the favor of doing all the heavy lifting. You will see these this semester. You will either work with them, you will generate them, or they will be provided for you. FASTQ files, you will see. Generating BAMs, that will happen. VCF files, they'll be provided because I don't think I'm going to take you all the way through the pipeline. That's not the plan for DNA-seq. But we are going to do part of it. There are pipelines that I also want to show you for this work, but we'll talk more at length when we get there. Lots of software that you're going to use as the semester unfolds, okay? Some of these things you've already heard about. So BWA, you're going to become very good friends with it. As you already know, you're going to get a quiz on at least the transformation, and you'll probably see it again at some point on an exam. And a final. Remember, a final is comprehensive, okay? So FASTQC, this checks the quality of your FASTQ file before you even work with it. The reason for that is, what do you think the quality of your data is going to be if your FASTQ is either corrupted or tainted or not complete? You're not going to get a full picture. So running that through FASTQC, it does you the favor of knowing that the quality of the data that you start off with is high. Illumina's FRED is now, what, 30, right? They were talking about going to 40, but they're still at 30. So if some of the FRED scores that you're seeing don't meet in that FASTQ, you need to find out what happened. But it's a way to look at it. SAM tools is a very important tool. It converts SAM to BAM, right? So as you heard, a SAM file is a text file, and the BAM is the binary version of it, smaller, easier to work with. But you have to get from a FASTQ to a BAM. It doesn't just happen. It's not magic. STAR we'll get into for RNA-seq when we move down the line a little bit, about halfway through the semester. GATK, you already heard me mention their best practices guide. This is going to be really, really, really important when we talk more about DNA sequencing, because this package is going to help you move through these files to get variant calls, okay? Picard tools, very important in our pipeline for DNA sequencing, whole genome sequencing, whole exome sequencing, because it does several things to those BAM files. It sorts them, it indexes them, it marks duplicates, so it's actually in a usable form that can now be turned into a VCF file. It's very critical as we move forward. VCF tools, I highly recommend that you read about these softwares, okay? This is gonna help you organize, manage, annotate, do all the things, bless you, all the things that you need to do to be able to work with a VCF file. You saw some of it. It's a large file. 99% of the time, it's not going to open with EndNote, EndNote++, or any of that. You open it, it just tells you it's too large. You're going to have to do it in your Linux environment to look at it. Anavar annotates these variants for you. Very good program. It's very, very critical for being able to identify what is the function. Okay, what if it's detrimental to what you're doing? What if it's a disease state? What if it's not? Okay, you need to understand what happens. Now, dependencies for the program, I'm going to wrap this up real quick. Some of these are Java-based. Some of them have other dependencies that have to be there. When you run FastQC, it's called a wrapper program. You're going to have to load cutAdapt, and you're also going to have to load FastQC to run trimgalore. But they all have to have Java before you do that, because if you try and load it, it's going to say no Java. So they're all going to be there together, and they'll tell you a lot of what's wrong, but they also won't tell you what's wrong. So some of the things you're going to have to read manuals. We will talk about this as we go through the active demonstration. There are differences between proprietary software and open source software. Some you pay for, some you operate yourself. But if no one's maintaining the system, if no one, for example, if you're a grad program and your lab says, I need a program on gene fusion, go write me a program. And you do it, and you put it in GitHub, and you release it, and it's open source, and it's out there. You graduate, you move on, and you never go back to that program. You don't update dependencies, and you don't update some of the coding on it. Think there's going to be bugs? You betcha. So there's problems. So we'll move on, and we'll talk about this when I show you more of the hands-on. We'll also talk about variation, because this is the key. There were problems with the original human genome. Okay, it was based predominantly on Caucasians of European descent. Is that the world? No. We have a very diverse world. So they really dug in to find other things. So we'll find out more about this. So your homework assignment, you don't have one yet. Don't panic. It's going to be a couple weeks down the road. What you're going to be given is 2 FASTQs from 1000 Genomes Project. I'm going to task you with checking the quality of it through FastQC. But I'm also going to task you with running it to get those trimmings done on it. Do you guys remember that he talked about trimming programs before in the last lecture, correct? That was the last lecture last week. We're going to come back to that, and I'll show you more about how to use this software. I'm not going to throw you to the wolves. This is hard, but you're also grad students at a medical school, so it's good exposure to learn how to generate the files that you're going to deal with. Okay? Ultimately, what I'm looking for is I want you to show me output that you check the quality of the FASTQ files. It gives you visualizations and HTML files that tells you that. And also, I want you to be able to get down to the SAM file so we can talk about what to do next. So that's what's coming. Y'all have an amazing weekend, and we will see you next week. I'd like to remind you before you go, my office hours are from 9 to 11. Please come see me. Shoot me an email if you're going to come so I kind of have an idea of how many are coming. I'm on the 5th floor, office 5025. Please have a great weekend.
Thank you very much. Have a good holiday. Great job. I like it.
Was that okay? I felt like I was moving too fast.
Well, that's my problem.
No, that's not your fault. I just want to know what's coming.
No, it's great. I mean, thank you so much.
Do I turn this off?
We need to.
Push.
Two.
Does it stay here?
Yeah.
Does it stay here?
Yeah. Just go.
Oh, here. Okay.
You want to just
One thing, I just started ironing this class. How, and I'm